In previous lessons, we've learned a little bit about the origin of life on Earth. At some location on Earth, perhaps down at these undersea volcanic vents, a living cell was produced from chemical reactions that occurred previously. The molecules that assembled into a cell included DNA. DNA, of course, is the critical hereditary molecule for life on Earth. It's able to store information, the information to build proteins. It can be duplicated so that reproduction can occur, and the DNA recipes can change over time as mutations alter the DNA sequence. This makes possible the evolution of living things. So we have a situation, perhaps 4 billion years ago, where Luca and its descendants were reproducing and filling up the oceans with bacteria-like cells. And they were evolving new proteins, for example, to survive in different environments. We've learned that a bacteria cell then is a cooperative effort between proteins with different jobs. Some proteins extract energy from food in the environment. Some proteins are building important molecules for the cell. And again, DNA stores the recipes to build all those proteins. In this lesson, we'll compare the early bacteria-type cells, which we will describe as a simple cell, with later cells that are more complex. So there is an evolutionary transition occurring. Perhaps around 2 billion years after the origin of Luca, cells are going to undergo a transformation. They're going to have new characteristics, and we will call them complex cells. One of the obvious differences is size. All these little rod-like structures here are bacteria. It fills the screen here. Those are our simple cells. These other two are complex cells. Now, not only are they much larger, but they do have other features that bacteria lack. So complex cells have a nucleus and structures called mitochondria. In this picture, again, we see an amoeba here and a paramecium. Those would be large, complex cells. And the little rod right there, circled there, would be a bacteria cell, a simple kind of cell. So we can see clearly the size difference, and we'll see that complex cells also have structures that bacteria lack. So here we have a cartoon drawing of a complex cell. Now, you might, in looking at this diagram, notice a few familiar scenes. We've got proteins attacking glucose, ripping it apart. We've got proteins building things. We've got transport proteins on the membrane. We have DNA. So this looks a lot like the bacterium. And in fact, there are many similarities, and that's no accident, because the complex cells are going to be evolving from ancestors that were simpler. And so they will be inheriting many of the characteristics of those simpler ancestors. So lots of similarities that we just pointed out. However, there are a few notable differences. The complex cells have this structure called mitochondria. There are many mitochondria in one complex cells, and we'll figure out what those structures are doing, but we can already see from the diagram it has something to do with metabolism of food. So the mitochondria will be involved in fully breaking down glucose to produce ATP molecules, chemical energy. But bacteria lack that. You'll recall bacteria have proteins on their membrane that are responsible for generating ATP molecules from the energy of food. For complex cells, that process happens in the mitochondria. In addition, the complex cells have a nuclear membrane. Notice both bacteria and complex cells have DNA. It's just that in bacteria, the DNA is just in the cytoplasm. There's no membrane surrounding the DNA. In a complex cell, there is a membrane surrounding DNA, and so the whole structure is called the nucleus. The membrane would be the nuclear membrane. In addition, you'll notice that the DNA of the complex cells is a straight chromosome, whereas in the bacteria, it's a circular molecule of DNA. Now here we see a picture of several paramecia, and the lighting is just such that we're able to make out the nucleus. So the blue oval structures there, those would be the nuclear membranes of each cell. Here we see an enlarged picture of that. So here's the nucleus of a complex cell, paramecia. So within the nuclear membrane here would be all of the DNA that the paramecium is using to build proteins. Now, all cells use this DNA information to build proteins. On the left here, we see our familiar diagram. This is how bacteria would do it. A region of DNA called a gene is going to be used to build a temporary copy, messenger RNA. The messenger RNA will hook up to ribosomes, which will then build the protein one amino acid at a time. The sequence of messenger RNA letters is a copy of the sequence of DNA letters. So DNA stores the information to build the proteins. Well, that's true in bacteria, but also in complex cells. So over here we have an amoeba, a complex cell, large cell with a nucleus and mitochondria, but we'll focus on the role of DNA here. The amoeba is currently making three important proteins, microtubules, choppers, and this membrane receptor protein. Each protein then is constructed in the same kind of way. There is a gene, a region of DNA that is the recipe to build that corresponding protein. Messenger RNA, in this case the red messenger RNA, is a temporary copy of the recipe for the receptor protein, and here we see the messenger RNA is hooked up to the ribosome, amino acids being linked together, folds up, there's the receptor protein. This gene is the gene for the microtubule protein. The messenger RNA detaches, hooks up to a ribosome, again amino acids are linked together, and this gene down here, the black messenger RNA, would be the recipe for the chopper protein. Notice the amino acid sequences is different for each of the proteins. The amino acid sequence is different because the messenger RNA triplets were different in the messenger RNAs. Messenger RNA triplets were different because the DNA sequences were different. So the amoeba is currently making three proteins, but in a process very similar to the way bacteria does it. It's just that the messenger RNA for the complex cells is being built on DNA within the nuclear membrane. Then the messenger RNA has to leave the nucleus to hook up to the ribosomes. In bacteria, the DNA is sort of just floating in the cytoplasm, and ribosomes as well are floating in the cytoplasm. So as the messenger RNA is being produced, ribosomes can go right to work. Here the messenger RNA has to leave the nucleus to hook up to the ribosomes. In this picture, again, we have a paramecium. Here's our cartoon paramecium. We see two genes then being activated. So messenger RNAs are being built. They have to leave the nucleus and hook up to ribosomes. So the blue messenger RNA is currently building this protein here, and the red messenger RNA is building this protein, and then the proteins go off and do their jobs. In this case, perhaps helping to digest food in a food vacuole. Now the messenger RNAs, after they hook up to ribosomes, they can be read, as it were, several times, and then the messenger RNAs will degrade. So they are temporary copies of DNA information. If the paramecium needs to build more of these proteins, it will have to undergo the process of messenger RNA construction again. So in other words, the cell has ways of determining when a certain protein is needed, when it needs to build more of a certain protein, and that's when the portion of DNA, the gene for that protein, will be accessed and used to build messenger RNA. And then the messenger RNA will be used in building that protein. Now, all cells duplicate their DNA prior to cell division. So we saw with bacteria, it's going to take its circular DNA molecule, duplicate that DNA molecule so as to give one copy to each daughter cell. And this is how bacteria reproduce. Well, so too with complex cells. They have to duplicate their DNA. Again, the straight chromosomes here. So the DNA will be duplicated, and then the nuclear membrane will have to break down so that each duplicate DNA molecule can get into the daughter cells, and then the daughter cells will rebuild a nuclear membrane. And that process is called mitosis, the nuclear duplication and division of the nucleus. But it's the same principle. DNA must be duplicated so that daughter cells can have a full set of recipes to build proteins. So for complex cells, as well as bacteria, we see the three features of DNA, the characteristics that make it a hereditary molecule. Information, duplication, change. DNA stores the information to make proteins. DNA can be duplicated for the purposes of reproduction, and sometimes mutations happen, and that produces change. So the offspring may be different from each other and different from the parents. Now natural selection will come along and operate on this variation. So factors in the environment may favor a mutation or penalize a mutation. So the differences that make a difference to survival are generated by change, feature of DNA, mutations. Now let's discuss the size difference between the two kinds of cells. Bacteria are very small compared to the complex cells. So while there are many similarities between the two types of cells, as well as a couple of notable differences, mitochondria and nuclear membrane, the size difference is another very big difference between bacteria and complex cells. So we've got a story then where the earliest cells, the first kinds of cells, were the smallest and simplest types of cells. But over time, in fact, for 2 billion years, bacteria evolved, but they never got larger. And then there was some important transition, some event that led to a dramatic increase in the size and complexity of cells on Earth. And we'll tell that story in another lesson. So to wrap up then, we do have lots of shared traits in the complex cells themselves. All complex cells are much larger than bacteria. They have a nucleus, which means a nuclear membrane surrounding the DNA, and the DNA is organized into straight chromosomes, not circular chromosomes. Complex cells have proteins that control the shape of the cell, and we'll see how important they are in future lessons. Complex cells are able to have a more active lifestyle. They can move, and key proteins perform those functions. And these complex cells have mitochondria, and we'll be learning that these mitochondria are involved in energy processing, extracting energy from food molecules and the production of ATP molecules that power the chemistry of life. Now, the fact that all complex cells have all of these features suggests a Darwinian inference. So when several species possess the same trait, Darwin considered the possibility that they inherited the shared trait from a common ancestor in the past. Well, since all complex cells have these features, size, and nucleus, mitochondria, cytoskeleton, etc., the Darwinian inference is that they inherited these features from a common ancestor cell which had these features. And in future lessons, we'll try to figure out how that ancestor cell came to be from its bacteria ancestors.